Hello?
Hi, can you hear me?
Yeah, I can hear you.
Oh, great. I'm fine, except I don't have a camera.
Oh, okay. You said you'd be able to present some slides today?
Yeah, I have a few slides.
Can you share your screen?
It says turn on caption, present now. How do you share screen?
Oh, when you want to share, you'll go down to the lower right hand corner. So present now, that button.
Okay.
And you'll click that button and you'll share your entire screen.
Oh, okay. So that would be good then, because since that'll work.
Okay.
That'll be better than the blank where my picture should be, perhaps. I left my camera at home, and I've been storm stayed, so that's why instead of driving right now, I'm sitting.
Yeah.
Yeah. Can you hear me down here?
Yeah. Yeah.
Okay. Well, good. I figured out the microphone at least.
Okay.
This computer doesn't come with a camera.
Huh. Is that a laptop or a...
It's a laptop, and it's a new one, and it's a gamer one, and it doesn't come with a camera. So I bought a little USB camera, and it seems to work fine, but I managed to leave it at home.
Oh. So you got a lot of snow?
Well, it wasn't that much. It was blowing all over the place, and I couldn't see. You couldn't see across the field. If I can't see across the field here, then I better not drive.
Yeah.
So about 4 inches of snow and really blowing winds.
Oh, okay.
That was Friday and Saturday. So Sunday, well, it cleared up. So I could have driven home on Sunday and then driven back this morning. So I figured that was useless.
Yeah.
Maybe not completely, but somewhat.
Yeah.
Waste of gas anyways. Home from here is about 100 miles.
Oh, wow.
Well, maybe more like 90 miles. Yeah.
Okay.
So yeah, not too good.
No.
So other people aren't joined yet, right?
Yeah, they should be coming at any time. I don't know. Sometimes they're a couple minutes late, but we'll just wait. See who shows up.
Okay, well, what have you been doing for research?
Oh, well, we're working on a couple of different papers, and we did the Basilaria paper, and we did a couple of other finishing up some papers on Bradenburg vehicles and things like that. So it's been a pretty diverse collection of things. Trying to do different, keep up on different things, and to do a lot of shifting back and forth between different topics. So, hi, Dick.
Hello. How you doing?
All right.
I don't have a camera.
Well, we got a good picture of you.
Hello, Vinay.
Sir.
Hi.
Yeah.
Oh yes, and there's a daycare in behind me here.
Okay. Good. Put some of the kids on.
Oh yeah, there's one in particular. Yeah, she would take over the show. She's good.
Hello, Devansh.
Hi, Bradley.
Hello, Yash. Let's wait a couple minutes for everyone to get in the room here.
Well, they're saying that why publications are like already excited to hear from that person, so they're not just booted another National Security Council. It's general ISIS. One of his
So how's everyone doing today? Anyone have any news or since our last meeting?
I can hear the daycare behind me.
Yeah, I can hear.
Is that a quiet activity, she says.
Yeah.
Then she balls them out. Oh.
Okay. All right, so welcome to the meeting. We don't have any, we have a couple things planned, but we don't have any talks today, like long talks. We have, I want to talk a little bit about GSOC, the Google Summer of Code, Yash. And then Susan is going to talk about the paper that she, we profiled late last year on embryos, and she'll talk about in more detail about that paper. We kind of glossed over it at that meeting, but she's going to give more detail about that, and then we'll discuss that. And then I don't know if Jesse's going to make it. He was interested in doing something, but I don't know. So let's see.
Okay.
Let's see.
My talk isn't much, but
okay, yeah, it's fine. Why don't I start with the GSOC stuff? Let me present. So, a little bit of news on this. The GSOC proposals are up on INCF's website. Let me show you this. So INCF is now running NeuroStars. It's something called NeuroStars. It's based on discourse. For those of you who know what discourse is, it's kind of like Slack, but it's a public forum that they have a special software that people can set up their own discourse. So NeuroStars is one of those discourse platforms, and INCF is using it for a lot of interactive communication for a lot of the people in INCF. So there are a lot of resources here. First of all, there are educational materials for neuroscience. There's a community encyclopedia, and there's standards and best practices, and they're all kind of hosted on NeuroStars, and people can comment on them and things like that. But one of the other things that NeuroStars is hosting are the GSOC projects for INCF. Yeah, Google Summer of Code is GSOC. So let's see. The first one, of course, is the pre-trained models for developmental neuroscience, and that's assigned to Project 14. And this is the description that we talked about in the meeting previously. This is through DevoWorm, so if you go, this is sort of, we'll have the community period through OpenWorm, which is, to interact with all the different groups in OpenWorm. So, for this project, there'd be like 3 weeks where you would present to the main OpenWorm group, and then, you'd have people from all the different sub-projects, getting exposure to what you're going to be doing, and then you might make some connections there. Usually we do the community facilitation period over Slack, and then we get into the coding after a few weeks. And so I have a couple of inquiries here. Here's one inquiry already where they're just kind of introducing themselves, and then, kind of getting them in touch with resources. So the first stage of this project is to have people submit proposals for the project. So, here's the description, and some people are asking some more specific questions about how to go about applying. And then once we get proposals in, and they go through maybe a couple rounds of editing on those, then we'll get to evaluate those proposals. So that's for pre-trained models. And again, it's just, a matter of them reading through this proposal. Then idea 15 is the OpenDevo cell integration. And of course, this is the one that we've discussed in the meeting that follows up on Vinay's work. And this is the description for that. And again, they go through the description and they can ask questions. So I have an inquiry here I haven't answered. People inquire about, whether they'd be a good fit. And usually they are, but, then you have to kind of steer them in the right direction in terms of putting together a proposal. So that's where the state of the Google Summer of Code projects. Probably be updating on that regularly. We have a couple of comments here. Vinay, I went through the GSOC ideas and they're interesting. I guess project 14 and 15 are related to OpenWorm. Yes. I don't know. I don't think OpenWorm is there are no other projects associated with OpenWorm this year. I made a couple calls and no one seemed interested, so I don't know if anyone's going to submit. I think you have until the end of the month to submit a project. Is 4D montaging of Axolotl image data fit? Yeah, we could maybe do that next year, but I don't know if INCF would sponsor something that's not explicitly has to do with the brain, but we'll see. I could propose it like under a different organization. So I have my own organization as well that I submit things under. Let's see. Well, yeah, I guess it would be to do with the brain. There's Jesse. Okay, here he is. Hello, Jesse. Is the formation of the brain. Yeah, I guess we could do that. Well, I mean, we can talk about it. That's probably too late for this year to get something together because I like to develop it a little bit. But probably next year we can do like something. Definitely, I think we can do it, though. Just you have to figure out what the tasks are and everything. So that's okay. So, Vinay said he gave me some leads and asked me to contact them. Yeah, I think this person here, I think, was from Vinay's university. So he did get. There are people who are kind of feeding into that. Okay. Yeah, Jesse says, I don't have anything to present today, but I wanted to stop in. Well, thank you for showing up, Jesse. So, yeah, we can talk about the Axolotl image data project maybe for next year or, figure out how to, what's all involved in that. But that would probably make a good project. It's just, we'd have to figure out what the tasks were on it and what we're dealing with on.
Yeah. My apologies for not sending you the pictures, the images of the axolotls yet. It's stuck in my brother's basement. And I do have the little disk drives for you, but so far I haven't been able to get down there for an extended period of time. Anyway, well, I'll try again.
What format would you be sending it in?
What are they? USB drives.
Oh, okay. I should
This
be
little
able to use
one.
them then. I just,
I got a. They were a sale, so.
Oh, okay. Yeah, I didn't know if they were like a floppy disk or something. I know that probably wouldn't be the case, but you never know.
No, I was debating sending a proper 1 terabyte drive, but these other things were on sale. I seem to be short of cash.
That's fine, yeah. Not that you're short of cash, but yeah.
Yeah, no, it was a bad farming year, so I have no farm income.
Oh, okay.
Yeah.
Okay. So, yeah. So Jesse, I see we're working on the paper this weekend, so thanks for that. I know it's been, kind of trying to find the time for that. So, yeah, that's fine. And you had some other ideas about what you wanted to present to the group. So, yeah, like the one you sent me a couple ideas. Those are all fine to do. We can do a short presentation on those. Speaking of which, maybe we'll do Susan's presentation now. Let me stop presenting.
Okay, well, I could start with reading something.
Okay.
I have something written by Adrian Williamson from physics.org. And it says, controlled hydraulic fracturing sculpts mammalian embryos into shape. And it describes better than the slide what's going on. So maybe what I should do is share my screen.
Okay.
Here, let's see. Present now, right?
Yeah.
So my entire screen sounds good, since I don't have. Oh, can you see my entire screen? No? There. That's better?
Yeah.
Okay, so this is just some papers
Actually, I don't think it's
discovered.
shared.
It's not shared? Oh, how do you get it to share?
Did you get, let's see, try it again. Go to present now.
So share your entire screen. Share, there we go.
Okay.
So it should be a picture of you.
There we go, it's starting now.
Okay. Wait. Does that look like hydraulics and hatching?
I don't see a screen. Let me see if...
I can see the screen. Hydraulics and hatching, early embryo dynamics.
Okay, who can see that?
I can see it just fine.
Oh, this one. Okay, I see. Yeah, it's a different tab here of mine. Okay. I can see it
Okay.
now.
Can everybody see that? Anyway, I was just putting this together, so I have, it's like this, and I've just copied and pasted from the source down at the bottom here. Can you see my mouse?
Yeah, it's moving around.
Okay, so there's the reference. And what you're supposed to notice is all these forced bubbles here that occur around I don't know what stage, about stage 32 or more, just before the blastocyst starts to form. And what they're saying is that because of the high pressure, this is like fracking in between the cells. Anyway, I'll read what this article has to say about it, because it describes it better than I do. It's a hydraulic fracturing or fracking is a process most often associated with shale gas extraction, but a team of researchers from the Institute Curie at Sorbonne and the Center for Interdisciplinary Research in Biology at Collège de France have concluded that cell fracking is the mechanism that moves an embryo, and here is a mouse, from a radial symmetric accumulation of cells to a bilaterally symmetric blastocyst. And it was published in the Journal of Science called Hydraulic Fracturing and Active Coarsening Position, the lumen of a mouse blastocyst. Anyways, it's embryonic self-fracting. Mammalian embryos use controlled hydraulic fracturing to sculpt their shape. So that's, I thought that was very interesting, and then I looked it up.
So this is for the mouse, right? Just for people who don't know.
So this is a mouse.
Okay.
But it's used as a model animal for all mammals.
Okay.
So it goes from the hydraulic fracturing here to a blastocyst.
Okay.
And you always wonder how the cells got stuck over on this side of things. And they think they have this explanation. Anyway, given that embryo cells are stem cells and that this is definitely high pressure, it'll do something to cause cell differentiation, or most likely. At least that's my thought on it.
Okay.
And if you've got high pressure inside of here, you're going to cause cell division, as is shown here, but I haven't gotten to the bottom of this slide. Maybe I had a couple of weeks ago, but I've been through some exams and stuff, so I just sort of, again, it's a copy-paste.
Yeah.
So it says multi-scale feedback interactions between luminal pressure and tissue mechanics control well blastocyst size. So they're saying that this controls the size of this blastocyst. It starts off smaller and enlarges. And from what I know of this is kind of like hatching contractions, where there is a zona pellucida membrane around it, and it has to get out of that membrane in order to implant into the mother. If it doesn't make it, it dies. And so this is a very critical stage. And people who do the artificial insemination stuff or implantation have taken a laser and tried to cut through the zona pellucida, and lately they've decided that didn't help that much. In other words, this embryo has to hatch. And here's some hatching contractions.
Okay.
And I read a paper about this quite a long time ago, and they said that in young mothers, they had too many contractions, and in old mothers, there were not enough, but just right, and it would crack the zona pellucida, and it would hatch out. And here's a picture of that in a mouse, although this is quite a wide crack. Like usually it's trying to slip out of a very tiny area. I guess I should have gotten one for contrast, but anyway, this is a lot of pressure as well. It's under pressure in here, and it leaves, squashing the embryo on the way by. And if they cut the zona pellucida off of the embryo and not let it hatch, it won't implant. There's something going on. So I thought that was interesting.
Yeah.
And I'm just kind of doing this. I'm not really opening it up. But anyway, so part of what's going on is if you have strain, the catalysts work better, and chemical processes work better on a curve. It's true in a mitochondria, and I assume it's true in an embryo. So if you have this type of thing going on, you've got more chemical reactions happening. And what else? Okay, this is just more strain causes binding sites for chemical reactions.
And so then, it's the strain that's actually enabling a lot of the biochemistry.
Yes.
Okay.
Yeah. The strain and the pressure would enable different types of chemistry as well. And then this kind of looks like Dick's cell state splitter. And so you get different cell adhesion when some of this is going on. Again, I haven't gotten back to this, so I haven't completed my thought about this, but there's definitely actin, myosin, and microtubules in play with this. And this is another slide showing that. So actin myosin network for force generation. And so the cells switch from being stationary and non-polar to polarized, and then they'll also switch to an active moving cell. When you've got mesenchymal cells, they move. It's a very important part of development again as the neural tube closes. And this is another diagram of how a cell becomes a moving cell. And again, this one, I threw this one in here because it looked interesting, but I haven't got to the bottom of it yet. So this is going to be really brief. And then this is actually some work that's done with Dr. Sharif. And he took this image, partial image of an egg blastocyst stage. And then again in his lab, his grad student took this image, and I believe that it's supposed to be more of an oval here. And here, this is a circle, and it looks like this is a mosaic cross egg, and they didn't develop. Or if they developed, then they swam in circles and died. So this is the wrong shape of a blastocell for this animal. And you can see should be extreme stress here, and we believe this is under a lot of pressure as well in here. So this is sort of backwards to a mammalian embryo. This is an amphibian embryo, and they tend to have the blastocell inside and the animal development on the surface, or on the surface here, instead of being internal. But again, pressure is involved. Or it looks like it is. So part of my task is to measure the pressure inside of the blastocell. It's sort of the beginning of some research here. But apparently I have to take a couple of courses first. Anyways, that's my complaint for the day. So do you want to see any more, or do you have any questions?
Please.
Well, I don't have any more slides. This is sort of halfway.
Well, let me go back. Let's see what's in the chat. Let's go through this. So Dick asked, Susan, can we have a copy of the slides? Yeah, you can send me this link to the slides or send Dick a link, but I would definitely like to have a link to the slides.
Okay. How would I send a link?
Is it a PowerPoint? You can just send the PowerPoint. You can attach it as an email, and
okay, I'll send it to you. All right, sure.
Oh, I was just thinking it was Google Docs, but it's okay. You can just send that as an attachment.
Oh, I can make it into Google Docs. I better not. This is an unpublished picture, and although they said I could have it, they gave me verbal permission to have this image. He's very protective of his images, so
okay.
Yeah.
And then Dick says, any evidence given for apical microtubules?
And these other slides? In these slides? They're just talking about actin and myosin in them. But yeah, it almost looks like there are microtubules in there. But they say it's actin. I don't know. This is just, I haven't investigated. Yeah.
Maybe they just didn't look for microtubules.
It's quite possible. Yeah.
Because I assume they're showing this up by fluorescence.
Yeah, I think so.
Yeah, in which case they would have to use tubulin, antitubulin.
Yeah, they would have to do that. So I guess then one could send them a note asking where the microtubules were in the situation.
If there are microtubules present, then this looks like a cell-state splitter.
Yeah, it does. I just like it was. That's why I plunked it in here. It looks like your cell-state splitter. At least this one does for sure. Yeah. But anyways, yeah. Just missing the stuff in the middle.
Well, the microtubules in a cell-state splitter can also be peripheral. Some cells, if you go back to the old paper by Golden and we showed examples where you can have around the perimeter of the cell both an actin bundle and a microtubule bundle.
Oh, okay.
Okay, so it doesn't have to go across.
Well, it looks like something's happening in the middle too here with some actin, which would make sense. Mm-hmm. Anyway, that's it's sort of half done slides, but I didn't know if I would have another good opportunity. I don't like being storm stayed over my weekend.
Yeah.
So, yeah, you never know; it might happen again.
Let's see, what other questions do we have? I guess Dick got another question. Local stress might initiate differentiation waves. So I guess this goes back to the slide with the cell-state splitter.
No, it goes to the Axolotl embryo with the optical coherence tomography.
Oh.
Yeah, because if you actually have a local spot of strain, that I think that's exactly the right place to get ordinary differentiation initiated. So it might be what's causing it.
Yeah, well, there is strain there, and this is under pressure. Well, you can kind of guess that it's under pressure because it's a perfect sphere. Yeah. But like I said, these are mosaic on mosaic eggs, and I believe they're slightly skewed, so to speak.
Again, if you look at papers, that would correspond to a stretch-activated contraction. The prediction would be that a contraction wave, that stretching might initiate a contraction, which then would propagate as a wave over the surface of the embryo.
Yeah, well, okay. That's kind of what I think too, that
It's nice to see the possibility of actually measuring the strain directly.
yeah, I'm hoping to get some images. Like I said, they're insisting I take a couple of courses first. I think he wants me to understand the mathematics behind the signal processing in the optical coherence tomography, because that's what this is. The blue is all optical coherence tomography, and he wants me to understand it. So I'm going, fine, I'm going to take your course. I won't guarantee I'll pass it, but I'm going to learn how it works.
Is Sharif teaching it?
Pardon?
Is Sharif teaching the course?
Yes, he is.
Oh, yeah.
So,
Okay.
yeah. So, and he's a good teacher. I really like him in a lecture. He explains stuff.
Okay, good.
Yeah. So I have to learn about it here, but definitely he's got a setup so that you can see what's going on. It's not as refined as maybe I would like, but they're trying different things in the lab. This is the one on the right here is a quadrature OCT, I believe. And it's a swept source quadrature OCT if you're into optical coherence tomography. And I'm supposed to be doing optical coherence elastography, which is another addition. So,
The elastography measures the local elastic property, so you're making a map of elasticity.
yes, yeah. So it's, you perturb it maybe with ultrasound, I don't know, so it shakes or bends, or you compress it a little bit and then it changes shape, and you can measure the before and after pictures to see how much it bends to look at the elastography of it. It's viscosity and elastography together. According to some of the papers that I've been reading, yeah, you get both. You've got all kinds of things like air puffs and piezoelectric squashers and, like I say, ultrasound shakers and working on all kinds of things. The elastography is certainly taking off. Like there's more papers last year than there was the previous year, and so on.
Yeah. Well, thank you for that. That looks pretty good. So yeah, I mean, I was just thinking about, like, if you go back up to the first slide.
Yeah. Okay.
Or the first one with content. Yeah, that one.
Yeah.
So they're actually putting in. They're introducing, like, forces here, gas bubbles or something. What is this?
Well, they're water bubbles under pressure. Yeah, they think it's water and it's under pressure.
Oh, okay. And
just
so
like
they're using it just to stimulate the in between the cells, and so.
Well, the embryo does this to itself.
Okay, but they're introducing more of it. Is that correct, or are they just observing this?
No, they're observing this from what I can tell.
Okay.
Susan, what are those bubbles?
The bubbles are water, as far as I can tell. Like that's what it said.
I assume they're surrounded?
They're rounded?
No, surrounded by membrane.
Oh, yes, they're in between the membrane.
Okay, but you see, if you take two cells that are adjacent to each other, they would have two membranes that were nearly touching. Okay?
Yeah, I can read the last part of this. I don't know if it is.
Well, no, let me ask a simple question. If you consider the membrane around a bubble, where does it come from?
Oh, yeah, they don't say.
They don't stay.
It's hydraulic fracturing. But it is under pressure inside the zona pellucida, so.
Yeah, okay, but I'm trying to understand, because I guess what I'm saying is the topology looks like it's tremendously disrupted. Okay, compared to just cells that are adjacent to each other with these little bubbles. So the question is, okay, the bubbles, they think, well, there's water inside. Well,
yes,
where does water come from? Because this is a section of a 3D structure, and therefore the water has to come from the cells nearby. And so does the membrane have to come from the cells nearby.
yeah.
For all these little bubbles. So it seems to me that waving one's hand and just saying that water-filled bubbles is quite inadequate description.
Okay.
Okay.
Yeah.
There's something lacking here in understanding how they're formed. Do they have any time-lapse showing the formation of these bubbles?
I'm trying to remember if they might have had a supplement.
Yeah, are these pictures live or fixed?
These are live. They're supposed to be live.
Well, they should be able to do time-lapse.
Okay.
So did you check their supplementary information, see if they give any time-lapse?
No, like I said, this is really half-baked, so I probably should check that out.
Yeah, look at the paper, see if there's a section of supplements with movies. If there are, it'd be great to see them.
Okay. All right. Well, I could send the paper along too.
Yeah.
Okay.
With this sort of introduction to this. It needs more investigation for sure.
Yeah, it's also 2013. Is that? Oh no, it was 2019.
It's a 2019. It's very recent.
Probably not much follow-up yet then. Okay.
Yeah.
It's a neat observation. And usually raises more questions than it answers.
Right. Well, this looks like things are under pressure.
Okay.
Yeah. And that's basically the idea, is that there's pressure generated at the membrane and it does things.
Stupid question. These are mouse embryos?
They are mouse embryos.
Okay, now remember for the axolotls, one of our criteria for keeping data was that the embryo developed normally.
Yes.
Do they have any? Now, with these, the being mouse embryos, you have to reimplant them into a mother and try to see if they develop.
Well, I thought that they were, they're pre-implantation, so they do a lot of work under a microscope with these embryos.
Okay.
If they're going to implant them into the mother. But yeah, did they implant them into the mother and did they develop? Exactly.
Do they have any proof that these embryos, which showed what they're calling fracting, actually were able to continue to develop normally?
I think that they followed up on that. Just like I said,
Okay.
This is too early for me to be presenting this, but it's the only time I can think of that I've got. So that's why I'm not sure.
Okay, fine.
I'll just include the paper. And yeah.
Okay, good.
Yeah, it's good. Thank you again, Susan. Does anyone have any other questions? I think that's it for the chat questions. But any other questions from Vinay or Jesse or?
No.
No, somebody's having a an argument in the background here.
Oh yeah. They said I heard no, and I was like, oh, I guess. So yeah, well, thank you again, Susan, for presenting. That was good. If people want to see the videos, you can go online to the paper. They usually have the supplemental materials there for people to view. And so then I guess the next question I have for people is, are people interested in presenting things in coming weeks? So we have our schedule. I know next week Yash and Devansh are going to be presenting on, I can't remember what the actual topic is, but they're going to be presenting something machine learning oriented. And we'll see, how that fits into all this. I mean, you guys can present and then we'll ask questions. And that'll be interesting. I don't know where Devansh is. He may be left early, but and so that's on the schedule for next week. And then the week after that, I'm not sure. I know Jesse wants to present sometime soon on some things. We can schedule that. We can schedule it offline even, like at another time of the day. And then we can replay. Oh, okay. Devansh is still here. And like I said, for Jesse, you can present at another time if it's more convenient, and then we can play the video of that in this meeting, just to give, make it more convenient for you. Okay, yeah, the replay thing, that would work. And you gave me a couple of ideas on different papers and things that you have of interest. Also, I suggested maybe, I don't know, I mean, you were the one who attended the Gary Marcus and Bengio debate. I don't know if you feel like you want to give a short recap of that at some point. Yeah, because I mean, that was interesting, sort of I think there's a lot of interpretation that needs to be done on that, so that's something to think about. And then Vinay might be interested in presenting something. Is there anything you've seen that's interesting, Vinay? Okay, he has some ideas for the second blog post on pre-trained models. So we talked about that before Christmas. That was the post that I don't remember what the actual topic was, but yeah, there's probably some follow-up to do on that. And again, like if you want to propose a blog post, okay, an extension of the first blog post, I think that's good. Well, so if you want to do that, then you can send me, maybe a draft, maybe an outline is more appropriate. And then we can talk about it in the meeting. Just, like in Google Docs or something. Yeah, this weekend. Just outline, hit some points that you want to hit. How do you want to extend this first post? What are the important points that we should extend upon? And then we can fill it in with sentences. And then maybe get some references as well so that we can do some further research and actually put some content in. Yeah, I mean, that would be great. I think, especially given the summer code presentation or the summer code proposals that are going to be written, I think having that second post would be good to sort of guide people on that. And then, of course, we can also do things that are more developmental. So we've mentioned that we're going to be getting the Axolotl data. So there's this data set that Richard and Susan have been developing, that they've been collecting data on. It's on the Axolotl, which is a very unique organism, has a very big embryo, which is why people are interested in it. And they have acquired a lot of data using the flipping microscope technique, which I think Richard presented on earlier. I think he presented it last week or last year, where they flip the microscope. They invert the embryo when it's under magnification, and you're able to see, the embryo at different orientations. And so Susan's going to be sending me those data, and then we'll be trying to figure out how to best represent the data. A lot of different ways computationally to deal with it. Nick says Axolotl was a lot like our amphibian ancestors. It's also like our amphibian ancestors. So what does that mean exactly? in the broader scheme of things. Know thyself. So yeah, we'll be probably talking about that in coming weeks as well. Maybe we'll just do like a session where we kind of think about like what we have in terms of data, and then maybe come up with some ideas on how to represent it. I know we have people here who are very well skilled at thinking about these things. So to get people's feedback on kind of how to go about making this happen will be, I think, fun. But again, if anyone has anything they'd like to present, if it's a paper or a topic, or even if you want to go over like an outline of something, please feel free to say I want to do it on this date, or I want to do this. I want to present on this upcoming date, and we'll set it up in the calendar, which I don't have open right now, but I can share that with people. And then we can we'll have this meeting this year. I hope that they're a lot more freeform than last semester or last year, where we did the presentations. We kind of had a presentation every week, but I hope that we can also get some good ideas going, maybe some new papers going. Then there's also a follow-up paper to the Basilaria paper. So the Basilaria paper, the first one, was the machine learning analysis of the morphology. We were thinking of doing a second Basilaria paper on Basilaria behavior. That one still kind of in the thinking stages right now. So maybe we can come up with an outline of that for maybe next week or the coming week after that. I talked a little bit, emailed a little bit with Thomas Harvick about that. So he's actually cultured Basilaria, and he's captured a lot of microscopy data on it. But we're still kind of thinking about that. So I know Dick had proposed that we do this paper, and I thought it was a good idea. So, again, this is something that'll be developing over the next coming weeks. So if anyone else has ideas about that, if you're interested in behavior in an organism and doesn't necessarily have any kind of brain, we can talk more about that. We have some things in the chat here. Jesse says, I'm not sure if embodied cognition would be better for this group or the other one, but I may do something on that. General definition, perhaps, like I'm working on in the manuscript. I think probably the other group would be better for that, at least initially. So we could do that in the other group, and then if it's really interesting to this group, we might talk about it here too. And then Yash says, wanted to clarify that we will be talking about some DL architectures, that's deep learning architectures, next week, for capturing spatiotemporal features from videos and sets of images. So it may be useful for the pre-trained models. Yeah, that would be very good. If you could tie it into the pre-trained models, or if you could remind us that it does tie into that at least, that would be a good discussion point. I think, I don't know much about DL architectures like that I can converse about right now. But yeah, so when you present, make sure that we're, as a reminder, we're a mixed group. We have people who are computer science people and people who have different backgrounds that aren't as computer science oriented. So, try to, if there's jargon, try to make sure that you sort of, explain it. Make sure you leave time to explain it to people, or, make sure that it isn't very, just like, cutting and pasting from a paper. Yeah, that's good, because it's, in general, that's a good way to do it, because you have a little bit, it facilitates a little bit more conversation. So Dick said here in this comment, bacillary needs simulations of alternative mechanisms. So this is for behavior. So yeah, for behavior, there are a lot of different things, different ways we can look at the behavior and sort of what's going on, how are they generating that behavior. And then we would match it to movies or data of the bacillary actually moving. And so, I don't know if people are familiar, but in the paper, Thomas does an analysis where he shows movement of the Basilaria colony. And there are these rod-like cells that are arrayed in a stack like this, and they move in a way that's, they move laterally to one another. So they generate these movement patterns that are quite exquisite for something that seems like a pretty simple organism. They fold kind of like an accordion. They move laterally across their edges, and so they generate these sinusoidal waves. But it's not just as simple as a sinusoidal wave. There's a lot of variation in there, and so we don't really know what the mechanism is that coordinates the movement. But that's kind of the idea of this paper, is that we would be exploring that aspect of it. But we have to, keep in mind that, we have to validate the data somehow. So, they have to basically be alternative mechanisms, but they can't be that outlandish. So Dick also says, Thomas is also generating SEMs, which are scanning electron micrographs that are spectacular and constrain possible mechanisms. So we actually have other forms of image data as well to work from, depending on what we need. So that'll be interesting. That'll be an interesting thing to look at as well. So I think Dick left for a minute here. And let me ask, does anyone have any other questions or things that they wanted to bring up in the meeting? Okay. Yeah, I think that's good. I think we have a couple of things that we'll be working towards, and hopefully everyone is. If anyone has any questions, you can contact me on Slack. You can email me, or just wait until next week, I guess. If you want to talk about something during the meeting, just let me know a little bit in advance. But otherwise, I think next week, so we have the DL architectures presentation. We won't have a lot of time for anything else. But how long do you think the presentation will be? We should probably aim for about 40, 45 minutes. It's usually a good time for presentations. We can make them shorter, but I just wanted to. Yeah, 40 minutes would be good. So 40 minutes will be good, and then we can discuss for about 20 minutes. And like Susan's presentation was pretty good. She had, she gave some images and some, a little bit of background in it. So now we have a good taste of what that research looks like. And then, you can also go into more detailed presentations. But, it's up to you. So. Well, anyways, thank you for meeting this week. It's been fun. And next week we will talk about DL architectures. And again, if you have anything you want to propose for some future week, we can schedule it in the schedule. And have a good week. Talk to everyone later.